Glorified be Allah subhanahu wa ta'ala. Assalamu alaikum viewers. Hope you guys are doing well. This is Zahra Qayyum. Today I'm gonna discuss about plasmid and its types. So let's get started with the plasmid introduction. The term plasmid was first used by researcher Joshua Lederberg back in 1952. Plasmid simply is an extracellular genetic element. However, some plasmids are attached to chromosomes. Although most plasmids are mobile or self-transmissible, the size of a plasmid can vary from 1 to over 1000 kilobase pairs. Plasmids frequently include genes that are necessary for an organism to survive, as well as those that are usually advantageous to the host organism, including antibiotic resistance genes. Plasmids are mostly found in bacteria, but they're also found in multicellular creatures and archaea. Then comes types of plasmid. Plasmids are classified into six primary classes based on their phenotypic characteristics. The first one is resistance or R plasmid. Mm, Resistance plasmids, by definition, carry one or more antibiotic resistance genes. Resistance gene contains the information for the production of a protein that makes an antibiotic ineffective and hence confers resistance against an antibiotic to a pathogen. The second one is fertility or F plasmid. F plasmid is characterized by the presence of F or fertility factor. It is also called the sex factor, which helps transfer genetic materials from one bacterium to another. Usually, the bacterium with the F factor transfers it to another bacterium which lacks the F factor, and the exchange happens through conjugation. The third one is plasmid. Cold plasmids contain the genes that code for colicins, also known as bacteriocins, which kill other bacteria. Proteins called colicins protect the host bacterium by eliminating other germs. Col E1 is found in Escherichia coli bacterium. The fourth one is degradative plasmids. These plasmids help the host bacteria break down substances including camphor, salicylic acid, xylene, etc., that are frequently not found in nature. Therefore, the genes for enzymes whose primary job is to break down particular substances are included in degradative plasmids. The fifth one is virulence plasmid. A bacteria that has a virulence plasmid becomes a pathogen or simply becomes an agent that causes disease. Bacteria that cause disease can travel quickly among people and infect a new host through reproduction. Virulence plasmids are found in Escherichia coli bacterium. Cryptic plasmids, the sixth category is cryptic plasmids. There is not a noticeable alteration in the characteristics of the host cell when cryptic plasmids are present. All they do simply is code for the enzymes needed in the host cell for their maintenance and reproduction. Based on the origin or source of plasmids, the plasmids may be categorized into two main groups: natural and artificial plasmids. Natural plasmids simply occur in nature. We don't have to make them in a lab. Prokaryotes and eukaryotes both naturally contain natural plasmids. Its example includes Col E1 plasmid. Artificial plasmids do not occur in nature, and they are created in vitro, means they are created in labs simply by recombining certain sections of two or more additional plasmids. These plasmids may be natural or synthetic plasmids. Its example includes PBr322 plasmid. Then we'll be discussing the Col E1 plasmid in detail, which is the example of the artificial plasmid. Simply, Col E1 is a multi-copy plasmid that naturally occurs and is derived from E. coli, Escherichia coli bacterium. Its copy number is around 40, with base pairs 6,646 base pairs. There are genes for synthesis of colicin E1 in the natural Col E1 plasmid. Colicin is an antimicrobial toxin generated in stressful environments. For using in molecular cloning experiment, these colicin genes are replaced with selection marker genes. Then comes PBr322 plasmid, which is the example of the artificial plasmid. A popular vector for E. coli cloning is PBr322. It contains 4,361 base pairs. The main constituents of PBr322 plasmids are as follows. The first one is REP, replicon region. The plasmid replicates itself through replicon, and it is derived from PMB1 plasmid. The second one is ROP gene, which produces ROP protein, rope protein. This rope protein lowers the copy number, and its source is also the PMB1 plasmid. The third and fourth are the tetracycline resistance genes and ampicillin resistance genes. Then We can see in the figure 1.1 PBR322 plasmid is elaborated. It contains two major regions, ampicillin resistant gene and tetracycline resistant gene, and the one is ROP region, rope protein region, which contains PBU2 and ORI. This site is auric site, which is origin of replication. Through this segment, plasmid can replicate itself. And the tetracycline resistant gene contains BAM-H1 and SAL1 genes, and ampicillin resistant gene contains PST1, PVU1 genes, and there is also ECO-R1 region, which contains CLA1 and HIND3 genes. Then comes PUC19 plasmids. PUC19 plasmids contain 2686 base pairs, and this plasmid is high copy number plasmid. It contains multiple clonic sites and a lactose Z sequence. The main components of PUC19 vectors are, the first one is PMB1 REP region, which is replicon region. The second one is BLA gene, BLA gene, which is taken from PBR322 plasmid. This encodes beta lactamase, which confers ampicillin resistance genes. The next is lacoperon system of Escherichia coli. This vector loses the rope gene, increasing the number of copies. This rope gene was present in PBR322 plasmid, but absent in PUC19 plasmids. As we can see in the figure 1.2, PUC19 plasmid contains ampicillin resistance gene, LAC-Z sequence, MCS, which is multiple cloning site sequence. It contains multiple genes, starting from ECO-R1 and ending to HIND3 genes. And it also contains ORI site, which is the auric site, origin of replication. Then comes TI plasmid. The plasmid was named TI plasmid, means tumor inducing plasmid, because it induced tumors in the host plants. The Agrobacterium tumefaciens bacterium carries the TI plasmid. The most important characteristics of TI plasmids are: this large plasmid contains 200 kilo base pair, has several genes involved in the process of infection. It contains 8 genes found in the tDNA, which are expressed in plant cells and give rise to malignant characteristics of the transformed cells. In tissue culture, this TI plasmid may grow as a callus forever, even in the absence of phytohormones, which are necessary for healthy in vitro cell development. TI plasmid has different regions with different functions. It contains tDNA, which is 20 kilobase pair sequenced, sequence boundaries, genes for opine synthesis, TRA genes, auric area, which is origin of replication, VIR region, which is virulence region, and TRA genes. As we can see in the figure, TI plasmid contains three main regions. The first one is virulence region, which is highlighted with blue color, and the second one is origin of replication, and the third one is tDNA region. tDNA region is the major region in TI plasmid. tDNA region contains two borders, left and right border, and contains auxin, cytokine, opine, etc. TI plasmid also contains open catabolism. And this was all about plasmid and its types. Thanks for watching the video.